Welcome all to AK Bio lecture series. In the previous class, we discussed that there are two mechanisms that regulate the cell cycle. One is external events and another one is internal events. We categorized the internal events into two. One is cyclin Cdk mediated events and another one is checkpoint mediated events. Today we will discuss the internal events mediated by cyclins and cyclin dependent kinases. Major proteins that control the events are cyclins and cyclin dependent kinases. We know that in the case of humans there are different cyclins and CDKs. Each have its own specialized function in different cell cycle phases. In human, different types or different kinds of CDKs are involved in the regulation of cell cycle. We discussed in the last class that cyclin D, CDK4 or 6 complex are essential for entry into the G1 phase of the cell cycle. When the cell is in the G1 phase of the cell cycle, the level of cyclin D is in optimum level to bind with CDK4 or 6. So at this stage, CDK4 or 6 cyclin D complex phosphorylate a protein called RB. RB is also called, the full form of RB is retinoblastoma. Actually, RB is a protein with multiple phosphorylation sites. First, during the early G1 phase, cyclin D, CDK4 or 6 complex hypophosphorylate RB protein by phosphorylating some of the phosphorylation sites of RB protein. Now, the RB protein is in hypophosphorylated form. The hyperphosphorylated form of RB inhibit the progression of cell into S phase by two mechanisms. One is hypophosphorylated RB along with HDAC. HDAC means histone deacetylases inhibit G1S transition. The histone deacetylase or HDAC is an enzyme that removes the acetyl group from the histone protein on DNA. And HDAC also recruits the histone methyl transferase enzyme to the DNA, making the DNA less accessible to transcription factors by heterochromatinizing the DNA. The second mechanism is hyperphosphorylated form of RB, that is hypo-RB, inhibit the protein called E2F, elongation factor 2. E2F family members play a major role during the G1 to S transition in mammalian and plant cell cycle. Then in late G1 phase, the cyclin E, CDK2 complex phosphorylate the hypophosphorylated RB again. The continuous phosphorylation at various sites of single protein can be simply called as sequential phosphorylation. The sequential phosphorylation transforms the hypo-RB into hyper-RB. At this stage, hyper-RB dissociate from HDAC and E2F protein. As a result of HDAC dissociation, histone acetylation occurs and heterochromatinized DNA turned into euchromatinized DNA. Now, the transcription factors can access the DNA because of its euchromatinized nature. Because of the dissociation of E2F from RB protein, that is retinoblastoma protein, E2F is free now to turn on the genes required for the G1 to S phase transition. Cyclin A CDK2 complex is critical in the S phase. In the S phase, cyclin A CDK2 complex is critical. Cyclin A CDK1 complex promotes the entry into the M phase from the G2 phase of the cell cycle. In the late G2 phase, cyclin B replaces the cyclin A, and the cyclin B CDK1 complex triggers mitosis. Now we are discussing how interphase to M phase transition is regulated in animals. Here you can see the points. Entry of a cell into M phase is initiated by a protein named MPF. MPF is a maturation promoting factor or M phase promoting factor. And MPF is composed of two proteins, a 32 kilodalton subunit called Cdk1 and a 45 kilodalton protein called cyclin B. These two proteins associated with one another to form the active MPF. In the last class, we saw that the cyclins and CDKs are evolutionarily conserved in nature, in structure and functions. Only a single type of cyclin-dependent kinase named CDC2 is present in yeast cells. Look at the points here. In the case of MPF, it is composed of the two proteins, 32 kilodalton subunit called Cdk1 and a 45 kilodalton protein called cyclin B. And Cdk1 is homologous to the CDC2 protein, that is CDC2 kinase of yeast. Among different CDKs in human, one that is part of MPF is called mitotic promoting factor is called Cdk1. We know the general activation process of CDK in general. The activation of each CDK is depending on its dimerization with the cyclin subunit, corresponding specific cyclin subunits. Here we are talking about MPF. Cdk1 is the subunit of MPF, and it will also be inactive unless it is bound to cyclin B. Cyclin B is a member of cyclin family, and cyclin B is also called mitotic cyclin. It is the concentration of cyclin B that causes the changes in the MPF activity during the cell cycle. Look at the graphical representations projected here. In the left side of the figure, you can see a model of the MPF protein. Here it is represented in two colors, isn't it? Cyclin B in blue color and CDK1 in green color. When you look at the graphical representation on the right side, you can see the same color representations. The green line parallel to the x-axis is the level of CDK1, and blue line, which is zigzag is the level of cyclin B concentration as time progresses. The concentration of both protein is given in y-axis. In the y-axis, you can see the concentration of both proteins. From the graph, you can easily understand that the concentration of CDK1 does not vary much during the different stages of the cell cycle. But the level of mitotic cyclin, that is cyclin B, rises from G1 and reaches peak level of concentration during G2 phase to M phase transition stage. When the cyclin B concentration rises in late G2 phase, they bind to form a cyclin CDK1 complex causing CDK1 activation and mitosis is triggered. Then when the cell exits its position from M phase into the next G1 phase, rapid decrease in cyclin B level will occur. As a result, the activity of CDK1 will also become rapidly reduced. Exit from mitosis and entry into next G1 depending on rapid decrease in CDK1 activity. Look at the graph here. In the graph, x-axis represents the stages of the cell cycle and y-axis represents the MPF activity level. From the graph, it is clear that during early G1 and late G1 or S phase stage, cells lack MPF activity. MPF appears in G2, early G2. MPF appears in early G2, rises dramatically in late G2 and reaches a peak in mitosis. Now you know that only cyclins bounded CDK is active and function its role in cell cycle progression. Isn't it? We found that MPF is inactive up to M phase. How it is maintained in cell is a curious question to answer. Now we are looking into the mechanism of CDC or CDK. In the case of fungus, it is CDC. In the case of higher animals, it is CDK. Now we are looking at into the mechanism of CDK activation and inactivation. There are 4 mechanisms involved in the regulation of cyclin dependent kinase activity or CDK or CDC2 activity. Which are they? First one, CDK phosphorylation and dephosphorylation. Second one, action of CDK inhibitors. The third one, controlled proteolysis. The fourth one, subcellular localization. First, look at the mechanism by which the CDK is regulated with phosphorylation or dephosphorylation. As in the case of any proteins, CDK and cyclin is also composed of amino acids, and each amino acids have its own property because of its variation in its R group. So amino acids such as serine, threonine, tyrosine take part in regulation of proteins by phosphorylation and dephosphorylation in eukaryotic animals. Actually, the process of activation and inactivation of any proteins is depending upon the position of these amino acids in the protein chain when they undergo the phosphorylation, and also it depends on the dephosphorylation activity. So, which type of amino acids and the position of the amino acid both are equally crucial, important in the case of regulation through phosphorylation and acetylation, etc. Actually, in the case of regulation of CDK activity, it is multi-step process of regulation. Before entering into the regulatory process of CDK, keep in mind that activity of 4 other regulatory enzymes, that is 3 kinases and 1 phosphatase enzymes involved in this process. Then one more point you must remember is that CDK, cyclin dependent kinase, have 2 inhibitory phosphate sites on tyrosine and 1 activation phosphate site. The activation site is on threonine residue. Let's discuss the regulatory process in detail. We will look briefly at the events that occur in fission yeast. We know that CDKs and cyclins are highly conserved in its function and structure from yeast to humans because of the fact that we all originated from a common ancestor. Look at the picture in the screen. In step 1, during G2 phase, first and second protein kinases called MYT1 and WEE-V1 come into the picture. That is, in the first and second protein kinases called MYT1 and V1 come into the picture. They phosphorylate a key tyrosine residue. Which amino acid? Tyrosine residue at the position of tyrosine 14 and tyrosine 15 respectively of CDC2 in yeast cells, and in the case of human the protein is CDK1, not the CDC2. In the given picture, the activity of MYT1 is not given. Tyrosine 14 and 15 are inhibitory phosphate sites of CDK1 protein. Means when these two amino acids are in phosphorylated state, the protein will keep on its inactive state. If these two residues are phosphorylated, the enzyme becomes inactive regardless of the phosphorylation state of any other amino acid residue of the protein. In step 2, during G2 phase itself, third kinase called CAK. CAK means CAK, Cdk activating kinase, that phosphorylates a critical threonine residue at the position of 161th amino acid of the CDK1 protein. Threonine 161 is the activator site of the CDK1. We saw two inhibitory sites also. Which are they? Tyrosine 14 and 15. But phosphorylation of threonine 161 by CAK is necessary but not sufficient for the activation of CDK1 protein. Due to the effect of MYT and V1 enzyme over the effect of CAK, keeping the CDK in an inactive state. In normal wild type cells, MYT1 and V1 keeps the CDK in inactive state until the end of G2 phase. Then, how this cell activates the CDK1 at the G2 to M phase transition state phase exactly at that moment? At the end of G2, the inhibitory phosphate at tyrosine 14 and 15 are removed by another enzyme, a phosphatase named cell division cycle 25. So the protein's name is that is a phosphatase enzyme, and its name is cell division cycle 25. This short form is CDC25. CDC25 activates the CDK1 cyclin B complex by removing the phosphate group from the tyrosine 14 and tyrosine 15 and triggers the initiation of mitosis. When the phosphate group is removed from the tyrosine 14 and 15 of CDK protein, the protein will be activated. Removal of this phosphate switches the stored cyclin CDK molecules into active state, allowing it to phosphorylate key substrates and drive the cell into mitosis. And the balance between MYT1, V1 kinase and CDC25 phosphatase activities determine whether the cell will remain in G2 or progress into mitosis. The second mechanism out of 4 mechanisms involved in the regulation of CDC is the CDK or cyclin dependent kinase is the activation of cyclin dependent kinase inhibitors in the regulation in the process of cell cycle. CDK inhibitors are also called as CKI, cyclin dependent kinase inhibitors, CKI. Cyclin dependent kinase inhibitors bind either with free CDKs or Cdk cycling complexes to regulate Cdk activity. So in budding yeast, in the case of budding yeast, a protein called SIC1, SIC1 acts as a Cdk inhibitor during G1 phase. During G1 phase of the cell cycle. Degradation of SIC1 protein, SIC1 protein, allows the cycling Cdk complex that is present in the cell to initiate DNA replication. Okay. In human, there are two distinct families of Cdk inhibitors, CKIs. Which are they? The INK4 family, INK4 family, and CYP family inhibitors, CIP and KIP. CIP or KIP family inhibitors. CYP proteins are capable of inhibiting all CDKs. There is no specificity. In the case of INK4, INK4 means inhibitors of CDK4. CYP means CDK interacting proteins, and KIP means kinase inhibitory proteins. INK4 proteins are tumor suppressors, and loss of function mutations leads to carcinogenesis. You can see the picture that representing different types of Cdk inhibitors at each phases of the cell cycle. You can see here. In this picture, you can see different names that of proteins that inhibiting the different cyclins at different phases of the cell cycle. And P16, P15, P18 and P19 are INK4 family proteins. Specifically inactivate CDK4 and CDK6 by forming heterodimer of inhibitors and CDK. So that function by forming heterodimer of inhibitor and CDK. Actually, the heterodimer formation prevents the dimerization of CDK4 or 6 with D-type cycling in G1 phase of the cell cycle and prevents the progression of the cell from G1 phase to the S phase. The P21, P27 and P57 are the CIP-KIP family of proteins form heterotrimeric complexes with cyclin D, cyclin E and cyclin A dependent kinase complexes. The CYP-KIP family proteins binds with CDK cyclin dimer and forms a heterotrimer complex. By this way, they inhibit the progression of the cell cycle. The P27 molecule incorporates itself across both subunits of the cyclin A CDK2 complex in S phase, form a heterotrimeric complex, then changing the conformation of the catalytic subunit of CDK2 and inhibiting its kinase activity. By this way, P27 inhibit the S phase progression. When P27 gets phosphorylated, it will be degraded, and as a result, the cyclin CDK complex will be free to progress the S phase. Absence of this protein in the P27 deficient animals allows the cells to divide several more times before they differentiate, that leads to cancer. The third mechanism out of 4 mechanisms involved in the regulation of CDK, CDC is the controlled proteolysis. So, in the regulation of cyclin dependent kinase, is the third mechanism is the controlled proteolysis. Degradation is accomplished by means of ubiquitin proteasome pathway. Degradation is an irreversible event that helps drive the cell in a single direction. It drives the cell in a single direction. Regulation of the cell cycle requires two classes of multi-subunit complexes. Which are they? First one, SCF, and APC complex is the second one. Both SCF and APC function as ubiquitin ligases. Then what is ubiquitin ligases? Ubiquitin ligases are the proteins that can tag the ubiquitination signal on the targets. We studied the action of APCs during the metaphase to anaphase transition in last classes, and SCF proteolyze the G1 to S cyclin, that is cyclin E. The proteins become targets for the SCF after they are phosphorylated by CDKs present in the late G1 as G2 phase of the cell cycle that regulate the cell cycle progression. The second class of multi-subunit complexes are APCs. The APC complex acts on the M phase and degrade a number of key mitotic proteins, including the mitotic cyclins, that is cyclin B. The level of mitotic cyclins, that is cyclin B, rises from G1 and reaches the peak level during G2 to M phase transition stage. Then when the cell exits its position from M phase into the next G1 phase, rapid decrease in cyclin B level will occur. Destruction of the mitotic cyclins allows a cell to exit mitosis and enter a new cell cycle. The final mechanism out of four mechanisms involved in the regulation of cyclin-dependent kinase or CDC is the subcellular localization. The cells of the eukaryotic organisms are elaborately subdivided into functionally distinct membrane-bound compartments. Subcellular localization is a dynamic phenomenon in which cell cycle regulators are moved into different compartments inside the cell at different stages of the cell cycle. Look at the graphical representation projected here. Here you can see the distribution of mitotic cyclin, that is mitotic cyclin B CDK1 complex in cytoplasm and nucleus. Here the x-axis is represented with the progression of the cell cycle, and y-axis is represented by cyclin B concentration. In x-axis, the stage of the cell cycle is started with G2 phase, and at that time the level of cyclin is maximum in the cytoplasm, not in the nucleus. When compared to the nucleus, it is maximum in the cytoplasm. But cell progresses into the prophase of the M phase. You can see a clear-cut change in the level of cyclin across the nucleus. At M phase, the cyclin B level is low at cytoplasm and high inside the nucleus. When a protein or mRNA is transported into and out of one compartment to another compartment, the proteins or mRNA must have a specific signal. When you studied the nuclear transport, you studied the NLS and NES, that is nuclear localization signal and nuclear exporting signals. Nuclear accumulation of cyclin B1 from the cytoplasm is facilitated by phosphorylation of one or more serine residue of its nuclear exporting signal, that is NES. Once phosphorylation has completed, the cyclin B can easily transport into the nucleus with the help of importing proteins. But the phosphorylation at NES, nuclear exporting signal of cyclin B, blocks the subsequent export of the cyclin back to the cytoplasm. That is the end of cyclin Cdk complex mediated internal regulation of cell cycle. In the next class, we will discuss the internal regulation mediated by checkpoint proteins. Thank you.